Hello everyone, and welcome again to Clinicals Explained. Today we're diving into a very important and commonly used test in the medical laboratory world, the Coombs test. The Coombs test, also known as the antiglobulin test, is used to detect antibodies or complement that act against the surface of your red blood cells. These antibodies can sometimes lead to conditions like hemolytic anemia, transfusion reactions, or issues during pregnancy. There are two types of Coombs tests, the direct Coombs test and the indirect Coombs test. Let's break down each one. Starting with the direct Coombs test, DCT. The direct Coombs test checks to see if antibodies are already attached to your red blood cells. It's commonly used in conditions of autoimmune hemolytic anemia and drug-induced hemolytic anemia. Here's how it works. A blood sample is taken. Red blood cells are washed 3 times to remove plasma and unbound proteins. Coombs reagent is then added. If agglutination or clumping occurs, it means antibodies are already attached to the red blood cells. This is a positive result. Coming to the indirect Coombs test, ICT. The indirect Coombs test is used to detect antibodies present in the serum, not yet attached to red blood cells. It's commonly used before blood transfusions and during pregnancy to test for antibodies that could attack fetal red cells, usually in the case of hemolytic disease of newborn. Here's how this works. Basically, this test is divided into 2 main steps, which are sensitization of the reagent RBCs, then the reaction with the Coombs reagent. First, the reagent red blood cells are sensitized by mixing them with the patient's serum. After they are sensitized, we wash them to remove all the unbound antibodies. Once washing is done, the Coombs reagent is added, which contains antibodies against the human antibodies attached to the red blood cell surface. That's why it's called anti-human globulin. If agglutination occurs, that means there are antibodies in the serum that could cause a reaction. Again, a positive result. To make this test possible, we need something called Coombs reagent, also known as anti-human globulin, AHG. You may be wondering how this anti-human globulin is made. Hybridoma technology is used for the production of these globulins. First, human serum containing antibodies, like IgG, or the complement, usually in this case C3d complement, is used. So the serum is injected into an animal, often a rabbit. The animal's immune system treats these human antibodies or complement as foreign and produces antibodies against them, specifically anti-human IgG or anti-human complement. These anti-human antibodies are collected from the animal's serum and purified, and that becomes our Coombs reagent. This reagent will bind to any human antibodies that are attached to red blood cells, helping us detect their presence through agglutination. Let's quickly summarize the key points. Direct Coombs test detects antibodies or complement already bound to red blood cells. Indirect Coombs test detects antibodies floating freely in the serum. Coombs reagent is produced by injecting human antibodies into animals, then collecting anti-human antibodies from them, and finally purifying them. Want to remember it easily? Think of the direct test as checking what's already on the red cells, and the indirect test as checking what's in the serum. Thanks for watching. I hope this video helped you understand the Coombs test better. If you found this helpful, please like, share, and subscribe for more medical lab tutorials. See you next time.